the cloud and I hit record to this computer and it just, I don't know where it went. It went into oblivion somewhere. So
Do they have like a video file?
no, you're not allowed to do anything to these computers. Like, so it was just pretend.
So I was like, you could probably find it.
No, it didn't write to the drive. So we'll start today with this second part, which I think is about transport. And this should be review for you. Let's call it 7.2, even though I know it's not 7.2 in the chapter, it's the 7.2 video, right? So we're going to start with nutrient transport. And I would remind you that transport is always about moving molecules across a membrane. And again, two basic types of nutrient transport. One we would call passive transport, and the other we would call, anybody remember? Active transport. Good. I did learn my lesson this morning. I had to keep moving active transport down the board, which was kind of funny because it is active. But today I'm just going to put it way down here. Put it over there. All right, what can you tell me about passive transport? Anything?
It moved with this concentration gradient, but from high to low.
So molecules, I'm going to put move with, we say, or down the concentration gradient. Which means from where there is a high concentration of whatever molecule it is to where there is a lower concentration of whatever molecule it is, right? Each molecule moves via their own concentration gradient. Because of that, why do we call it passive? No energy required. Yep, and energy in cells is in that form of ATP, right? And I would again remind you that when we talked in chapter 8 about why cells need ATP, what they do with it, right? They do those three things. They do chemical work, transport work, which is what this is going to be, and mechanical work, right? So of different categories of transport work, we have one that we would call simple diffusion. The molecules that can diffuse all by their lonesomes without any help from anybody are predominantly gases. And of course, the big two would be carbon dioxide and oxygen, right? But other things, other gases diffuse as well. Nitrogen gas would diffuse, carbon monoxide gas would diffuse. I would hope that you've spent a lot of time in anatomy and physiology talking about partial pressures of oxygen in lungs or in capillaries and why the oxygen diffuses from the higher concentration to the lower concentration. And the carbon dioxide diffuses the opposite direction, from where it's a higher concentration to a lower concentration. Yeah, well, hopefully you will soon, right? If I say this, facilitated diffusion, what's necessary for facilitated diffusion? Yep, of course, you gotta have some sort of a protein. It's a transmembrane protein or a protein that goes through the membrane, an integral membrane protein, right? So it's usually sort of shaped like a channel. It has a hole in the center of it so that things can move from one side of the membrane to the other. Sometimes you'll see them called channel proteins or carrier proteins, right? And they're going to help move a molecule, but still with the concentration gradient. Those are usually a little bit larger molecules. Think like glycerol. Or maybe part of an amino acid, right? Like a carboxyl group, right? It's not really a whole organic molecule, but it's bigger than carbon dioxide. Probably the most famous of passive transport, our friend osmosis. And osmosis is the movement of water, right? Across the membrane from a higher concentration of water to a lower concentration of water of the same molecule, right? I would remind you that in most solutions, the water is the solvent. And the molecules that are dissolved in the water or in the solvent, we call those the solutes, right? And the concentration of the solutes is what's going to affect the concentration of the water. Just have to be sure that's not Australia. Okay, it's not. Good. Alexa, I think. I heard yesterday, he said he was going to go to class and then go play a soccer match. So I was like, I thought you were there for school. That's what's most important. But I'm far away, so I don't really know what he's doing, but he likes it. There's no resolved be there. Right. So I'm going to have to draw the 3 beakers. I'm sorry. But I'm compelled to draw them. And into those 3 beakers, right, I'm going to have to put some water, a solution. If my solution is 100% water, like super pure water that's been double distilled and deionized, right? And I have some little bacteria down here. I'm making them our friends E. coli. Right, I have some bacteria down here, and in their cytoplasms, let's say they have about 80% water because they have other stuff inside their cytoplasm, right? They have proteins, they have ions, they have sugar, glucose, all kinds of stuff in there. Where is there a higher concentration of water? Not volume of water, but concentration of water. Outside the cell or inside the cell? Outside the cell. So where will the water go? Inside, right? So the water is going to move this way. What will happen to my cell? Let's let it swell first. Right? A cell will most likely swell because it's a cell, right? Most cells that are constantly exposed to this water differential, many of them have ways to get rid of the excess water that comes in so that they don't burst. Right? But in the most extreme situations, yes, there could be swelling. There's definitely going to be swelling, and there could be lysis of the cell. Right? I'll put it on the list. What kind of a solution is this? Hypertonic? Isotonic? Hypotonic? You didn't vote. Okay, we'll do it again. Hypertonic? Isotonic? I vote all times so as not to sway your opinion. Hypotonic. There you go. Bingo. Hypotonic. Yes. All right. That's a hypotonic solution. The solution where I make it exactly perfect, where I make it 80% water and 20% dissolved stuff, like let's say salt, glucose, phosphates, whatever, right? The solution that I make it just perfect. Again, like Goldilocks and the 3 bears, right? This one's just right because inside my cell, there's about 80% water. Now, where will the water go? Nowhere, they say. Well, yes, it will go one in, one out, right? It will be equally balanced, right? What will happen to my cell? Yeah, cell will stay the same. What kind of a solution do I call this? Isotonic. Oh, good, you got that one right away. Yeah. I know, it's the other ones that are hard to remember. Pick one. I don't care which one, and remember that one. And then just figure out the other one if I ask you that one. All right, so then last but not least, what if in my last beaker I have like 50% water and I have like 20% salt and 10% glucose and we're up to 30%, okay, 20% other stuff, solutes. And I have my little bacteria in here with their 80% water inside of them in their cytoplasm. Now, where will the water go? Outside the cell, because it's not the volume. This might be 100 milliliters, right? Or a whole liter. Obviously not inside the cell. I have very small volume, but the water will move from inside the cell to outside the cell. Now what will happen to my cell? It will shrink. Very good. And depending on how great of a differential that is, it may die. Draw like a little sad face. Why do we care? Why do we have to do this again? Because two reasons. Because you still mix them up. That's one reason, yes. Because I am on an endless quest to try to help you learn things that you were supposed to learn before and then actually remember them for the next class that you're in. I'm on an endless quest for that. As well as in microbiology in particular, I want you to think about what we talked about the last time we were together about the media, right? And if you looked at any of those formulations in your book between then and now, you will notice that the amounts of stuff that you put in is very, very small. Like you might put in 2 grams of salt or 1.5 grams of something or 0.000 grams of a particular amino acid. And that's because that media that we are making, we make it all in a liter. And we try to keep it as isotonic as possible, right? Because the whole goal of making the media is to make the bacteria grow in the laboratory so that we can study them. And if we make the media too dilute or too concentrated with what we think are good things, right? Too nutritious, we're going to kill the bacteria and then we won't have anything to study. Right? So that's like one practical reason why we have to go over this again. The other has to do with food preservation, because I would remind you, thinking again about Tuesday when we all went downstairs and got pineapples and strawberries and virgin pina coladas, right? Thinking about food preservation, if you look in any pretty much food, any culture you want to pick, right? For thousands and thousands of years, people have been preserving food with salt. I want to call it salt, you want to call it spices, whatever you want to call it, right? Or sugar. You eat jam, jelly, maple syrup. Yes. She's like, right. So that has so much sugar in it, you can practically leave it out on the counter and no bacteria are going to spoil it because it is so hypertonic. I forgot to write that. It is so hypertonic that the bacteria can't live there. Fungus, yeah, they'll start to lay their spores down and grow through it, but bacteria, pretty much no, right? I would add to this list vinegar, right? If you think about like native foods that people eat and try to preserve their foods, like I said, I don't care where in the world you're thinking about this, salt / spices, sugar, and vinegar are the 3 big preservation, and especially if you combine the salt or sugar with the vinegar, right? Make some pickles or some kimchi, whatever you're mixing up, right? Some borscht. I'm thinking about Ukraine and Russia, right? So whatever you're mixing up, right? Sauerkraut, right? it has those things in it. The vinegar is the pH changer, right? So we're going to talk about that in another day, right? Or another time, or you can watch the video about that. But just sort of in general, I want you to think about it because that's how we preserve food, basically, even bread. A lot of the additives that are in bread or in meat that people are like, oh, don't have meat with nitrates. Okay, have meat with listeria. Go ahead. That's the point of the nitrates. It's not just to make the meat look red, it's to kill the bacteria that would poison you, right?
So it's the sugar and the honey that keeps that from ever spoiling?
Yeah. Yeah. And maple syrup too. Not quite as, not as much, but like people refrigerate it more than they do honey. But I think it's again for the fungus. Right. Okay, I think that's good for passive transport. Let's talk a little bit about active transport. Right. So what things do I have to write about active transport? It requires ATP. And why is that? Right, because if the molecules are moving, it's not working. Against, we say, or up the concentration gradient, which really means in terms of concentration from where there is a high concentration. No, sorry. Where there is a low concentration, right, toward where there is a higher concentration already of that molecule. So you can think about active transport like stockpiling, right? We've already have some of this, but we just keep bringing more in, right? Examples of active transport, usually we think about pumps, like our friend, the sodium potassium pump, right? Again, if you've already had this in A&P, you'll remember this is about maintaining a membrane potential. So sodium and potassium are moving via simple diffusion all the time, but your cells actively pump them in and out to maintain a particular minus 70 millivolts or whatever it is, minus 90 millivolts across the membrane so that we can transmit electrical impulses for things like muscle movement and neurons, right? Another type of active transport that is specific to bacteria is called group translocation. No. This is prokaryotes only. Kind of cool thing. I'm gonna have to draw that. So, right, we've got ye old phospholipid bilayer here. It's similar to facilitated diffusion in that it involves a protein that spans the membrane. Purple for proteins. Right, some more phospholipids. But the cool thing is, it will only transport a specific molecule. So there's a protein involved, like a protein carrier. It is specific for one molecule. And the thing that it does is when the molecule gets inside the cell, it gets chemically modified. So I'm going to put chemically modifies the molecule, so that's IES, so that it's different on the inside and now it can't fit back out the same hole, basically. The most common modification, part of the reason why it's in active transport, is because the most common modification is adding a phosphate, taking a P off of ATP and sticking that phosphate on the molecule. So get a different, go back to my orange friend here. So let's say this is glucose. Oh, we know glucose from chapter 8, our favorite chapter in microbiology. Right, so this is glucose. Glucose can be transported through to the inside here. I'm not drawing all the hydrogens and oxygens because I'm lazy, right? And then an ATP is sitting in here. It's going to add one of its phosphates to the glucose, and then obviously it's going to become ADP. We'll put it, have the yellow sun. But now this glucose, it cannot go back out the same protein. And like I said, this is unique to bacteria. Eukaryotic cells don't do this. They bring things in differently by bulk transport. So that would be number 3 on the list. These words again, you may know bulk transport. I'm going to put here mostly eukaryotic cells. Mostly your cells and fungal cells and protist cells and algae cells will do this, not bacteria so much. But we include it because, right, just like we put this one on here, that's a bacteria only thing, right? We want to keep reminding you all the different types of organisms that can cause disease. So these are your old friends, pinocytosis, right? Phagocytosis, yes? Heard these words before? Exocytosis for waste removal. And my personal favorite, receptor mediated endocytosis. Yeah. That's long to write, which is why we just abbreviate it RME most of the time. So pinocytosis, this is bringing liquids in, right? We usually tell you it's like sipping, meaning a cell is going to suck in a whole bunch of liquid or mostly liquid. And in that liquid, they're going to bring in also some nutrients that are needed. Something a little bit bigger, like a glycerol or part of an amino acid would be a good example of things that we brought in. Or if you're a eukaryotic cell, there's a lot of pinocytosis in your small intestine because that's where your food is actually absorbed, right? The nutrients, the little bits, all the way down to glucose and the proteins and the amino acids are actually absorbed. And so some of those are small enough that they could just kind of be brought in with a bunch of liquid. Phagocytosis, we think of particles, right? So this sipping the liquid particles / food. And I'm going to put as a reminder, right? If I'm a protist, for example, a bacteria might be food for me. I'm an amoeba, for example, right? Or a paramecium. And I run into a bacteria, I bring it in, and that's a big piece of food to me, right? And I'm going to digest it. Remember, the lysosomes are going to fuse with the phagosome and break everything apart. The waste, that's what the exocytosis is for. Waste removal on a cellular level, right? In a vesicle. These are all in vesicles. Receptor mediated endocytosis is also going to utilize a vesicle, but a very a different type of vesicle, such that the lysosome does not usually fuse with the RME vesicle. So I'm gonna erase this a little bit, draw you a little picture there, give you an example. So in receptor-mediated endocytosis, we have to have a receptor. The receptor is a protein. It's on the surface of the cell. It is specific. Not quite as specific as an enzyme, but it has a place where a particular thing binds. The thing that binds, we give that a name too. We call that the ligand. So the ligand, that's the molecule that fits in the receptor. And just like with an enzyme, unless it's a pretty close fit, it's not going to bind, right? That's kind of the minimum of what I need you to know about receptor-mediated endocytosis. I'm going to draw some other things here and talk for a minute about like a specific example, not because that specific example is going to be on the test, but to kind of give you some context for why we're talking about this, okay?
What is the ligand again?
So the ligand is the molecule that fits into the receptor, right? So the generic, like, you know, analogy for BIO221 students, since it's March, it's basketball season, right? I would say something like the 5 men or women who are on that college basketball team on the court at that moment, or the women's softball team here at Camden County College, they are going to be specifically designed and they have practiced forever and ever to catch basketballs or softballs, bounce them, throw them, right? That's what they do. Could they catch a football if I threw it at them? Yes. Hockey puck? Sure. Soccer ball? Yeah. But that's not what they're really built to do, right? That's not what they're really supposed to be doing. It's a little better as a football analogy because there are actually players on the field that are only allowed to be receivers, right? Unless you're playing rugby, right? But if you're playing American football, there are people that are designated as receivers and they're the only ones that can catch the ball, right? And they're looking for a football. And again, could I throw a hockey puck at them, a softball, a baseball, a basketball, a soccer ball? Sure. Would they catch it? Probably. Reflex, right? But what they're really looking for is the football. And only the football, right? So the ligand is the ball. Now, sort of example, but again, to give you a little context, because although this is not cell biology, it's worth talking about for a minute. So here's a cell, nucleus, nucleolus, right? And this cell has phospholipid bilayer here, right? Embedded in that phospholipid bilayer, there are going to be receptors for things. And they do actually occur in more than one place on the cell. Like a cell doesn't have just one receptor sticking out there for whatever it is. This particular receptor is for so this particular receptor, we're going to make it specific for epidermal growth factor. So the ligand is EGF, which stands for epidermal growth factor. Right? And the receptor is actually called, oh, I could have made that purple. Right. The receptor is actually called it's actually called that. EGF dash receptor, R for receptor. Right? So when epidermal growth factor goes floating by in the cell is not going to sip this or bring it in bulk because it's a small protein. It's going to bind to the receptors. And again, as you might remember from BIO1, these receptors are going to clump together And we're going to get invagination of the membrane here, where we're going to have lots of these receptors that are bound to epidermal growth factor. And the cell's going to be like, oh, this is something we need. Let's bring this in, right? So this is going to be put in a vesicle inside the cell. And I'm just going to draw one of my receptors with the epidermal growth factor on it. Right. And this cell gets a special coating. I'll make it black. This vesicle, rather than being a phagocytic vesicle, it gets a protein that gets put all over it, actually way back here when it starts to form. And that protein is not important, you know its name, but that protein serves as like a protection against the lysosome. So that the lysosome doesn't come over and just obliterate this thing, because the cell is going to recycle the receptor, put it back out there, and it's going to take, this vesicle is actually going to split into two, and one of them is going to have just empty receptors in it. That's going to head back out here to the membrane to be stuck back out there again. And the other one is going to be full of epidermal growth factor. You're like, where is this going? It's going to the nucleus, because we're almost to the punchline. Epidermal growth factor is a transcription factor. So what it does is it binds to promoter, which remember is a particular piece of DNA, to attract RNA polymerase so that transcription can happen. And transcription, remember, is to make messenger RNA. And then that messenger RNA is going to be translated into a different protein. And this protein takes an action. It tells the cell, hey, time for mitosis. Right? So I'm trying to help you like put all the things that you're learning into context. And like I said, this is not a cell biology course, but I think it's useful every now and then to remind you like sort of how the whole cell works, right? In terms of a eukaryotic cell. So let's just say, right, I'm 4 years old and I just had a growth spurt and now I have bigger bones and bigger muscles. I need more skin, right? Because we can think of epidermal growth factor, epidermis, right? Even though epidermal growth factor actually also stimulates bone development and muscle development, but that's a separate story. Go to cell biology, learn all about it, right? But sort of very basically, if I have more bone and muscle to cover, I need more skin to cover that muscle and bone, right? And because I picked epidermal growth factor, and most of us know that the epidermis is what is our skin, right? That's why I picked that one. It's going to go to transcription. It's going to get into the cell, and it's going to go as a messenger to the nucleus to say, hey, it's time for mitosis. It's time to undergo replication of all of our DNA and turn into two cells so that now we have more skin, right? So that's why receptor-mediated endocytosis is so powerful, because it's just for one particular molecule that has some other function. It's going to trigger some events. Usually they're transcription factors. So usually it's going to trigger particular proteins to be made that then do something in the cell or for the cell. All right, it's enough of that. Let's talk about some environmental factors that influence bacteria with microbial growth. So I would say that all the things we've talked about so far, or most of them are chemical factors, right? If you want to think about carbon and nitrogen and hydrogen as chemicals, right? And now, and transporting these chemicals around inside the cell or in or out of the cell. And then now we need to spend a little time talking about what other environmental factors influence bacterial nutrition, bacterial growth. Looks like my orange shows up erased either. All right, so I think let's do easy one. Temperature first, right? So that's probably the first one in your book, yeah. So every bacteria has an optimum temperature that it likes to grow in, but there's overlap of where it will still grow a little bit too cool or too hot. We put bacteria into categories based on what temperatures they like. Psychrophiles, not psycho, psychrophiles with an R, right? These are cold loving bacteria. So I think, let's say like from minus 15 degrees Celsius to maybe like plus 10 degrees Celsius. I'm sure the numbers are different in your book. It's not super important. It's not a hard and fast number. Looks like they're going from minus 20 to plus 15. So it's fine here. Minus 20 to plus 15. Try to remember that your temperature is 37 degrees Celsius, freezing is 0, right? So where would these bacteria live in the wild? Ice, good. Where would that be in the world? Antarctica, Greenland, Finland, Iceland, right? North, yes, right? Where else is it really, really cold? Down in the deep blue CCC, yep. Deep sea, so that's where you would find them. Where would you find them in your home?
Freezer.
Yes, in your freezer or your refrigerator.
I'm gonna say in my house, it's freezing in here.
Well, probably not actually in your house, but freezers are 0, like to minus 20 degrees Celsius, right? And refrigerators are about 4 to 6 degrees Celsius. So they would fall certainly in that area, like listeria. That's where listeria lives, in your refrigerator. Well, it comes to your home usually in either from contamination or from like uncured meats, unpasteurized milk, right? So, and it like for you to get listeria is not horrible, it's not pleasant, but you could get antibiotics to treat it. But if you are a pregnant person, it can cause stillbirth. So it's really important not to eat like goat's milk during that, a lot of it during that time, unless you can pasteurize it somehow, or at least warm it up. Mesophiles, what do you think they like? Moderate temperatures, yeah, the middle, middle. Let's see, psychrophiles, psychrotolerance, mesophiles, green. Looks like they've got about plus 15 to 50. And then again, where would you find them in the wild? Pretty much everywhere. Sun-warmed soil gets to that temperature, right? Compost piles, swamps, bogs. I live in New Jersey, so we have some of both of those. Right? What about thermophiles? What do you think they like? Yeah. And I think the regular thermophiles we put at 50 to 80. And those also could be in a compost pile, peat bog, thermal vents maybe. Let's say hot springs maybe. And then the extreme or hyperthermophiles. They're above 80 degrees Celsius. So those would seriously be really hot springs, volcanic vents. Again, this is an important thing to consider if I'm trying to grow a particular bacteria, right? If that bacteria is a let's say it's a chemo autotroph that likes to live at 70 degrees Celsius, then I'm going to need to provide it with sunlight and any other of its chemo autotrophic needs, as well as a hot temperature that's way hotter than the incubators that we have, right? I can't just go out in the wild and collect those and bring them back and give them nutrient agar and say, grow, I want to study you, right? So temperature, pH is probably next. Actually, your book does oxygen next, gases. I'm going to do pH next because oxygen is a long one, so we'll do a short one, a couple short ones. As we've said 100 times before, most cells, most organisms live between pH 6 to 8, but that is generality, right? There are bacteria that we call acidophiles because they love acid, right? They love low pH. So again, they would live places like sulfur pits, where it's very acidic. Also, your stomach, right, where it's like pH, what, 1, 2, something like that. These will be the bacteria that could spoil foods that are preserved with mild acids, right, eventually. I have to tell you this good pH story, right? Because again, in order to grow the bacteria that you want to study, you have to provide them with the right conditions. And if you don't have the right conditions, you might miss a bacteria that is normally growing there. So there were some guys, some physicians that were studying people who they were gastroenterologists. They were studying people who had ulcers. And they were treating them. It was like the late 90s, late 80s, early 90s, actually. They were treating them with antacids predominantly, right? To raise the pH in the stomach so that there wouldn't be this degradation of the stomach acid causing the ulcers, because it's what everyone thought caused ulcers, was the acid produced. Some people had over producer acid cells in their stomachs, right? Yeah, I know those cells have a name, intrinsic factors, that was called something maybe. Anyway, so these guys were studying ulcers and they were treating their patients, and they literally happened upon a patient that they had treated. That person was coming back. They were getting the endoscope done, when they stick down, look inside their stomach, and their ulcers were really cleared up, and they were like, oh, what's been going on? And the patient said, oh, I had a UTI and I just took a round of antibiotics. And they were like, oh, that's weird. And so then they started asking other patients whose ulcers seemed to magically get significantly better between when they saw them first and when they saw them last. And they started accumulating this sort of anecdotal evidence that people who had been on courses of antibiotics, their ulcers were better. And so they were like, maybe we're missing something here. So they took some fluid, let's say, from some patients' stomachs, and they managed to culture some bacteria in an acidic broth. They named them Helicobacter pylori. They grew them up, and then they were like, huh. They did a total Dr. Jekyll and Mr. Hyde. They drank the cultures, gave themselves ulcers. So they had scoped themselves first. They were all clear. They gave themselves some Helicobacter. Back down. Now I have ulcers. And because they were physicians, they could just prescribe themselves some antibiotics. And then they did it again, and the ulcers went away. And then they went to a scientific meeting that was probably sponsored by, like Pfizer or Zantac or something, where they were selling all these bicarbonate drugs that were increasing the pH of people's stomachs, right, to more basic conditions. And they presented their results to a silent room. People were just like, what? You can't do that. I'm like, well, they were like, well, we couldn't like do a study on people, right? We couldn't say, hey, you want some ulcers? So we were like, we have to find out somehow. And you know how hard it is to do little things on mice like that? Like, and you can do lavage techniques on mice in their lungs and their stomachs, but you can't, like, there's no little tiny endoscopes that you can stick down an anesthetized mouse to like look and see if they have ulcers, right? So yeah, they completely changed the whole world and how people thought about how you get an ulcer and how you get cancer. Actually, some Helicobacter, long-term Helicobacter pylori infections with some of the strains of Helicobacter can cause pyloric cancer, right, in your pyloric sphincter from a long time. Yeah. So be careful if you're looking for a particular bacteria, right? You have to give them the food that they like at the right pH and the right temperature, or they won't grow, right? And no one had seen that for a very, very long time. Yeah, now they're quite famous. Yeah.
Like H. pylori overgrowth is pretty common, right?
Right. And then you get a course of antibiotics, and a lot of times then it doesn't come back. Right. So, but before in the 80s, we would have just fed you a bunch of tablets to make your stomach, your stomach less acidic, which also then would affect your digestion of some particular proteins, blah, 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 right? And not really make the bacteria go away. Well, people take like people's species now too as that's another thing. I mean, like stomach contents, I would take that before I would swallow a pill with someone else's fecal bacteria in it. Well, if you had C. difficile that they couldn't treat with any antibiotics because it was multi-antibiotic resistant, and someone said, if you take this, it'll go away, you might. People do. I mean, it's a thing.
I tried a couple antibiotic regimens of flagyl first, and then take it 5-6 times. If it don't work, then
No, that's to kill protists. That's not to kill C. diff.
that's what we treat all the patients with C. diff with flagyl.
That kills Giardia.
Well, someone thinks it kills flagyl.
Ah.
That's what all the doctors prescribe.
Yeah, that was found accidentally too. It was basically found in people who had up the other end, right? So it had colonoscopies and the equipment wasn't being cleaned well enough. And so what was happening was people who had chronic, who had to come in for repeated colonoscopies because they had irritable bowel or they had a C. diff that they couldn't clear up, accidentally were inoculated with some healthy person's bacteria because they were just at 50 and getting their first colonoscopy, right? And then they were cured. And again, it was kind of an anecdotal observation at first, and they were like, wait a minute. And so then now there are regimens where they recruit people who are healthy people, and they like collecting blood, right? Taking your blood, like blood for bucks. They take your feces and they separate out the bacteria and they grow them in culture and get colonies going of all of them, and then they put them back together in a capsule. I mean, obviously there's not any fecal material in there anymore. They put it back in a capsule, and then people can take the capsules, and they're full of normal flora that can repopulate your intestine. Yeah, that's a thing. I don't just make this stuff up, as much as it sounds like it sometimes. I'm not making any of it up. All right, let's talk about oxygen requirements or gas requirements. It's really about oxygen. And this is a little tricky because it's kind of a two-factor idea that we're talking about. I need you to think about oxygen being required for metabolism. Chapter 8, again, as the final electron acceptor, right, on the electron transport chain to make ATP. Right? We know that story. Aerobic respiration, right? Now, the part of the story that we didn't talk about before, or didn't talk about very much before, is that, remember how I drew the picture of the proteins in the membrane, right? And then the ATP synthase, kind of like that, right? And the hydrogens going through, right? And I said, some of the protons join with oxygen here as the final electron acceptor to make water. Remember that story? Right? And I said others of the protons get recycled so they can be pumped again, and we can do this over when the next NADH comes. Right? Sound vaguely familiar? Yeah. Well, I wasn't telling you the whole truth, of course. When aerobic respiration is working at top speed, sometimes there's more oxygen with extra electrons than there are protons being pumped fast enough through ATP synthase to meet up with that oxygen and form the water. And we call those toxic forms of oxygen. And one of those is something called superoxide free radical. Another one is called oxygen permanganate ion or something. I don't remember. It's not important. I'm just giving you that one as an example because we're going to talk about the mechanism that's in cells to help prevent. So these toxic forms of oxygens like superoxide free radical, what that does is it causes mutations. It causes damage to the DNA. Which can lead to mutations. Try to remember, if I'm a bacteria, I probably only have one copy of all of my genes. So if they get damaged or mutated, I might die, like in the next 20 minutes, right? Us as people, we have 2 chromosomes, right? We're dihybrid, no, not dihybrid, what's that word? Diploid, that's the word I'm looking for. All right, we're diploid. We're diploid, so we have 2 copies of everything. So even if one copy gets a little damaged, we have a spare most of the time, right? So this is more dramatic in bacteria and more immediate in bacteria. Now, cells that use oxygen or use aerobic respiration, they have a mechanism to get rid of this superoxide free radical. Okay. They have an enzyme, it's called SOD. And what SOD does is it turns superoxide free radicals into hydrogen peroxide. And yeah, hydrogen peroxide. Yeah, that's okay, except this is also poisonous. So we need to get rid of that. And there are two enzyme systems to get rid of that. One is called the catalase enzyme, ASE. And what catalase does is it's going to turn that hydrogen peroxide into water and molecular oxygen or gaseous oxygen. Harmful, lovely, not dangerous, not toxic. There's also peroxidase. And there's no oxygen produced in that one. It's just two water molecules. And I'm not showing you the chemical reactions on purpose because that's not what's important to me. What's important to me is you understand that if I am an organism that uses aerobic respiration, I must have a way to get rid of this superoxide free radical. Because not only am I producing it while I'm making ATP, which is kind of a disadvantage, right? But it is also in the air from other organisms that do aerobic respiration. So even if I'm a bacteria minding my own business and I don't use aerobic respiration, there's still a low concentration of superoxide free radical around me in the soil, in the water, in the air. And if I don't have a way to get rid of it, I die. Literally. Yeah, it's bad. So SOD stands for superoxide dismutase. Superoxide dismutase, SOD. So stopping the mutations or the mutagenicity of superoxide free radicals. That's why you eat broccoli, right? Which sort of, I guess.
Why would broccoli?
Broccoli is an antioxidant. Okay. Broccoli helps destroy free radicals. You have to eat a lot of broccoli. Probably more broccoli than you would want to, but whatever. So now we're going to talk about oxygen requirements. And I'm going to tell you that you can group the bacteria in groups based on whether they use aerobic respiration and or whether they have this enzyme system to get rid of the toxic forms of oxygen. Okay. And obligate aerobes and obligate anaerobes are the two main categories on the far extreme, and then there's one in the middle that we call a facultative anaerobe or anaerobes. So obligate, must have, obligation, must do, right? And so an obligate aerobe is a bacteria, a microbe that requires oxygen for its metabolism. Most of them would be positive for SOD. That's hard to test for in the laboratory, but it's really easy to test if an organism has catalase. What happens when you put hydrogen peroxide on your skin? It bubbles. It bubbles is the right answer. That's what bacteria do when you pour hydrogen peroxide on them. If they have catalase, they bubble. And then you know they have SOD, and you know that they are probably obligate anaerobes, obligate aerobes. All right. Obligate anaerobes must, AN, not have oxygen. All capitals, must not be even exposed to oxygen because they do not have SOD and they do not have catalase or peroxidase. These facultative anaerobes, these are the fun ones. These are the ones that can do aerobic respiration or ferment, right? They usually, but that's why we call them facultative anaerobes. They usually grow, we say grow better with oxygen They will continue to grow, just more slowly without it. And they do have catalase.
I have a question about the obligate anaerobes. So my cat once got into a fight with a neighbor's cat and got trapped in the shoulder and got an infection. It's horrible. We had to take it to the vet. It ruptured in my hand on the way to the vet. And they go in, they clean out all of the goo. And then they left his wound open. And they said that it was because of the bacteria that could form. They wanted to leave it open. Is that why?
Yeah.
Huh. He is gone.
It is disgusting.
He lived, he's fine.
He lived. That's good, because soon all the people in my stories die, and then my students are always asking me, do you have any stories where the people live? And I'm
They
like,
eventually
no.
started out.
They all
He was
die.
as hateful as ever.
Right.
He's a neighborhood sheriff.
That's fine. All right, now almost the complete opposite of a facultative anaerobe is an aerotolerant anaerobe. So I'm going to put them in here, aerotolerant. And that means just like it sounds, right? They will tolerate oxygen, free oxygen. But my recollection is they do not have SOD. I'm going to have to cheat and look. They have alternate mechanisms for breaking down peroxide and superoxide. So they would be SOD negative, catalase negative, or peroxide, but they use an alternative method. It's not installed that. Alternate method to break down, right, hydrogen peroxide and superoxide. And then last but not least, I would put on this list what we call the microaerophiles. And I think they only have SOD, so they have part of the pathway. But they live where there's only a little bit of oxygen, micro, smaller.
What does it say? Yeah, I can't, we can.
This one? Micro, okay, well, I'll put it over here. Microaerophiles. So love, right? Love files, loving a little bit of oxygen, micro amount, microaerophiles. And like I said, they will grow. Like I said, they don't have at normal atmospheric conditions of oxygen, but they can grow where there's a little bit less oxygen. Now, the cool thing is there's a picture in your book that shows, so what you can do is, if you remember last time we talked about reducing media that you can make that absorbs the free oxygen in the media and in the tube, right? So you can make that reducing media and you can use a needle and stab different bacteria into that media, and they will only grow where the concentration of oxygen suits them best. So you'll have bacteria that will only grow at the bottom of the tube where there's little to no oxygen. Some that'll grow all the way through, like the facultative, right? And the aerotolerance will grow a little bit lower, but not completely on the bottom. The microaerophiles will be like a scum on the top. And the obligate aerobes also will, the microaerophiles will be a little bit under the surface, and the obligate aerobes will only grow on the top. So you can see what an organism's oxygen requirements are. So think again about my story about if I'm trying to collect bacteria from the environment and I don't really know what I'm looking for and I don't really know what they like best, I might want to do something like this, right? As well as change the pH of the food that I'm feeding them, right? As well as change the temperature, and to see what grows, right? Because they're going to have different requirements than just in the body. Like when we talked about the 5 eyes at the beginning, it was like set the incubator at 37 degrees because that's people temperature, right? But just, you know, thinking a little more broadly, you might have to make different choices. They show you an anaerobic chamber there. There's a person who's in like a anaerobic hood. So she's working in a hood where the oxygen has been vacated. It's a plastic screen. The gloves are built into the screen, obviously, because she has to breathe, right? And if you look at the picture real closely, you'll see there's a little incubator off to the side of her. And that it would be an incubator where you pump in carbon dioxide. You can't like completely get rid of all the oxygen. But those aerotolerants, a lot of the bacteria that are human pathogens are really, they grow better when you give them an aerotolerant environment. And so if you pump in like 5% CO2, which is like tissue concentration of CO2, they'll grow better than if they're just in a regular incubator. There's also a jar that you can get. It's called a brewer's jar because of brewing things like alcoholic things. And basically what it does is it grows organisms without oxygen. You put a little compartment in there, an envelope that has a chemical that will suck the oxygen out, the free oxygen. So you put your bacteria on the petri dishes, throw them in the jar, add water to the pouch, stick it in there, and then clamp it all down. And it's anaerobic until you open it. So that kind of works. Cheapy version, you can get a big old, like a restaurant size glass pickle jar, and you can put your bacteria in there on petri dishes, stub of a candle on a piece of cardboard, light the candle, close the lid, seal the lid. When the oxygen runs out, the candle goes out, right? Now it's anaerobic-ish until I open it again, right? So that's kind of like the cheap man's way to do it. And then there's expensive, you know, buy the special containers and have a whole special hood for it. Those of you that have some experience in the hospital, when they draw blood cultures, for example, they draw 2 tubes or 2 bottles. One goes in a regular incubator and one goes in an anaerobic incubator without oxygen, because some of the bacteria that you might be looking for are anaerobes. All right, that I think kind of ends what we need. Yeah.